Hello, and welcome to the DNA 2.0 webinar on protein engineering via the systematic exploration of sequence space. I'm Laura Whitman, Director of Biotech Marketing for DNA 2.0, and I'll be your facilitator for this webinar. Before marketing, I was a research biochemist in both academic and pharmaceutical labs. We'll start in just a minute, but first a few housekeeping items. Feel free to ask questions throughout the webinar. We will do our best to answer when possible or at the end of the webinar. You can submit your questions by typing them into the question box on the right side of your screen. If your question is not answered during the webinar, we will email you an answer within the next 24 hours. Second, we are recording this webinar and it will be available for viewing on our website in about 24 hours. You should receive an email with the link. I'd like to thank you for joining us and hundreds of researchers from around the world to listen to this webinar. At this time, I'd like to introduce our speaker, Dr. Sridhar Govindarajan. Sridhar is a DNA 2.0 co-founder and serves as our Chief Information Officer. He leads our company's automation and protein engineering efforts and has more than 20 years of scientific computing experience and research. Thanks, Sridhar.
So, hello everyone, welcome to the webinar. My name is Sridhar Govindarajan. And today we will cover aspects of ProteinGPS technology that we have developed here at DNA 2.0. to engineer proteins. And the title of today's topic, as Laura pointed out, is Protein Engineering via Systematic Exploration of Sequence Space. We offer this protein engineering as a service to our customers, along with other engineering solutions like the vector GPS, pathway GPS, and genome GPS. But what's common is the underlying systematic exploration part of the sequence, whether you're looking at codon optimization, amino acids, or pathways. And in all of these technologies, we couple them with machine learning approaches to help us identify what's the best sequence. And I hope you really enjoy this talk. So first of all, I would like to start off with a brief review of current approaches adopted by protein engineers everywhere. They can be broadly classified into two categories shown here. One I would call the rational approach, and the other empirical, the rational approach using structural analysis, understanding of the mechanism which enzyme acts, and an empirical is often based on libraries that are synthesized, and the emphasis here is a lot on diversity, selection, and screening. The first approach, what everyone calls as a rational approach, often based on thermodynamic and steric principles. It requires a structure for the enzyme obtained via crystallography or some sort of homology model-based structures. In this approach, you can see the calculations are often approximate. No one understands the physical-chemical forces to the atomic level, so we use approximate energy functions, but also they are very computationally limited. Since it cannot be done on a typical desktop. Nowadays, people use Amazon cloud services and harness thousands of computers to do calculations. And they primarily be able to predict binding instability. There have been a lot of examples showing success in this area. But also recently, there have been many papers that have created novel activities based on a natural scaffold using computational approach. But these do require further engineering. And in this approach, the idea is to identify amino acid changes based on some sort of a structural model, and then go about making new proteins based on these changes. Typically, you measure somewhere between 5 and 25 different newly synthesized proteins and test them. Okay. That's a big advantage of this approach, which is basically looking at a few clones to test, but also you are relying on something, on calculations that are approximate. The second approach shown here is what we call empirical protein engineering. It uses principles from natural evolution of enzymes. Okay. So the idea is to make many variants via mutagenesis and recombination. You generate a library and then you screen, find those with better properties through some sort of an artificial selection system that one has set up. and you pick the best, and then you make more variations, more recombinations, and another library, and you iterate this till you find the best clone. The advantage of this approach is there is no mechanistic understanding required. Nobody needs to know the structure, how actual mechanism of the underlying reaction is, and so on. But what it does depend on is surrogate assays, or high throughput assays as we call it. So it has a heavy reliance on high throughput measurements because you're screening lots and lots of clones, and you are relying on the fact that these surrogate high throughput measurements actually reflect your real activity of interest, or what you really care about. And I will emphasize this point quite a bit in the coming slides. And right away, I'm just going to have you another slide to illustrate this point, which is oftentimes, I'm not exaggerating here, when you're engineering therapeutic enzymes, you try to do that by screening through some sort of a fax machine for binding, and then you hope it works on mouse. Okay. So there's a big problem with these surrogate high throughput screens that they're generally very poor predictors for activity defined assay. But if you do have a nice assay in high throughput that will reflect perfectly what you care about, then obviously this is the best method that you can choose. Okay. And after the talk, I'm going to give you a little tool that I'm going to introduce you to, which you can use to play around with on our website. And that tool allows you to look at how data get transformed when you have noise, or data get transformed when you are actually looking at a surrogate assay. So this will give you a better feel when you take a look at it. And I will show a little demonstration of that tool at the end of this talk. Okay, so now we have a problem, and what is the solution? And one can simply think of this, okay, I have a simple solution, which is, why don't we just measure what we care about instead of measuring something indirectly? So why don't we go and optimize the function directly in a low throughput assay? But there's still a couple of problems associated with that. One is, oftentimes these assays are very expensive, so you cannot measure a lot of clones. Secondly, such assays may also be associated with a lot of noise, because you're looking at measuring something in plants or in fermenters, which are big systems and maybe hard to control. So along with this challenge, I'm going to list the three primary challenges that we face today as protein engineers. One is the sequence space is huge. If you look at 100 amino acid protein, in each amino acid, you can have 19 other options. The space is massive. So what do we change? How do we go about doing this? If you do random mutagenesis, you are exploring a very, very minuscule fraction of the space. And a lot of the space is not functional. Mutations are deleterious, for example. The second is assay costs are high. As I said, if you set up a low throughput assay in plants or in fermenters, then your costs are very high for every clone that you want to measure. On the other hand, high throughput assay also comes with costs in setting up initially systems that are quite expensive. And third point is the data is very sparse and noisy. So if you do mutagenesis and you pick two clones, they are completely different. There is nothing you can learn from that. The mutations are often not common. That's what we call as sparseness in the data. And data can also be very noisy. So our goal is to see how do we map this space. Okay, so that's where we started thinking about and developed what we call as ProteinGPS technology. So it's primarily designed to address the issues we saw in the last slide. And let's break it down one by one. Okay. First is the sequence space. Instead of searching randomly through all possible sequences that you can obtain from your starting point, one can limit yourself to what sequences evolution has tried out. So it's what people call as knowledge-based analysis of sequence space. Okay. So our goal is to reduce the sequence space to search by limiting ourselves to functional space using simple evolutionary models. The second one is systematic evolution. Instead of generating random libraries, we can specifically look for sequences in this space that helps you learn. Okay. By this process, we can actually limit the number of sequences to test and thereby reducing the assay cost, for example. So you can limit yourself to low throughput assay but still be able to understand what the space is. And this also partially solves the sparseness in the data problem. And the third is the learning, which is basically using this sequences that we have generated in the sequence space to test, how can we map those sequences and understand what's going on. I'm going to go over these in more detail in a few minutes. Okay, so now this slide gives you an overview of our entire protein GPS approach. I'll cover through each of these steps and how we do things as we go along. But let's start with initial starting point. This is the most important part of the problem. Customers or protein engineers typically have an enzyme that does some activity. Let's say it converts a compound A to B, and it shows some amount of conversion. It may not be specific, and the kcat km may not be very high. So what we want to do is to take this and engineer this protein so that it matches what you would want commercially. So there are two ways to go about it. So you can look in literatures, find proteins with known activities, and customers have in-house samples that they have screened, and you have a starting point. And there's another option too that we offer as a service. Okay, so I'm going to so let's assume that you either have a starting point or you have an enzyme that fairly reflects what you want but needs to be engineered. In one case, what we do is we do what's called optimized phylogeny sampling. So if you have in order to get the best starting point from nature, we can go back and take your starting sequence that we have and search in databases for all the homologs. And given the sequence information that we have in various databases, usually a wealth of information there available, we usually can pull out 200 to 10,000 sequences that are homologs of your given starting sequence. It's not realistically possible to go and collect each of these genes and test them. On the other hand, we don't have to do that. So what we can do is look at them and find out related sequences and pick a representative set from, let's say, this part of the tree, and so on and so forth. So you can then sample this tree by only synthesizing and testing a few extant sequences. So DNA 2.0 has developed technologies to be able to express these proteins in a system of interest, so they can be basically transformed from a real sequence or a virtual sequence in a database to what you get in a tube, and we can test these sequences for the activity of interest. So then once you have that data, you can use that information to say, hey, which one of these is the best and optimal starting point for my engineering of the protein. So this here you can have several properties coming into consideration. One is simply, let's say you're interested in high kcat over km, and second, you want to look at proteins that are more soluble, even if they have slightly less kcat over km. Sometimes you may be looking at thermostable enzymes, and so on and so forth. So this will give you a rough idea of what is your starting point going to be. Based on exact requirements, we can pinpoint what we want to start with and move forward. So that part really is about getting to the starting point. Where do I start searching the sequence space? Once we find that, then we do an initial design to take that sequence space and find regions of the sequence space that we want to sample. So here there are two parts to it. Okay. One is we have a starting point now, and we need to have a choice of substitutions that we want to make on the starting point. And this is critical because a lot of literature have shown that a majority of mutations are going to be deleterious and a whole bunch of mutations are going to be neutral. And making random changes is going to deal with more deleterious mutation than what you really like. So we want to minimize the number of dead clones that we're going to snap. So how do we choose substitutions? So we take the protein of interest, and then we go again look at all the homologs from the tree, and we usually get a multiple sequence alignment from that. So this already has a wealth of information that we can use. It will provide you with positions that you don't want to change at all, like these glycines here, because nature has never attempted to change that position. So that takes out some substitutions that you can possibly make. And also it gives you certain patterns. Certain substitutions are seen more frequently. Certain substitutions, one can do some evolutionary model and identify that they are adaptive, and so on and so forth. So by looking at your starting sequence and looking at the alignment, we can rank order all possible substitutions that are seen here in this alignment and assign them a adaptation score based on whether a position was conserved, based on whether how many different amino acids are seen in the given position, and so on. So we start with the sequence, and then we look at the alignment, and then we find out all the substitutions you can make, and we rank order them. Typically, when you do so, what you find is a Gaussian distribution of adaptation score for all possible substitutions. And if you look at a protein alignment, you will probably see more than 2,000 possible changes you can make from your starting sequence just based on the alignment. And this is not random changes, these are changes that are seen in homologs of the protein of interest. So what we do next is we want to choose. It's not realistically possible to take all 2,000 and explore that space. We want to be smart about it. We want to minimize the deleterious mutations which are probably lying around here, and we want to maximize the mutations that are more likely to affect my function in a positive or neutral way. Okay? So we choose top 57 substitutions for this example that I'm going to show here. And the number 57, I'll explain that later, but it could be top 30, top 100, whatever that. And for that starting point, we want to constrain ourselves to looking at only this space of 57 sequences. That sequence space is already massive. You have a starting sequence, and you have 57 possible substitutions you can make. The sequence space is 2 to the 57. That's already 10 to the 17. And making a library is not an option here, because you are going to sample, even if your screening size is 10 to the 7, about 10 to the 10th of this space. And such library option would not allow you to learn about the sequence space either. So what do we do? What we do is we design a small set of variants that we call infologs. And these are designed and these 57 substitutions are systematically varied to cover the space with the goal of learning from them. Okay? I'm going to give you a little more definition of what we call infons. And this term was coined by us. You won't find them in literature. But what you do find in literature is the term homologs. What are homologs? You can typically people refer that as naturally occurring genes that share common ancestry and trait. So if two genes are homolog, which means at some point back in time they share a common ancestor. But what are infologs? That's slightly different. What they are is they do share ancestry, but in the immediate past. We start with a gene and we make it we independently design synthetic genes that are derived from that starting gene, where substitutions are systematically incorporated. Okay? You were looking at the sequence and you were scanning everything around it in a very systematic fashion. So each sequence in an infolog set would be equidistant from each other and would be about 3-4 changes from your wild type. So you can call this as a synthetic ancestry, and with the goal to learn around the sequence space around the sequence of interest. So for our project here, what we will typically do is you have your starting gene. So we found the sequence from the tree that we would like with the right properties. We found 57 substitutions from the alignment that are very useful. And now what we're going to do is to design a set of infologs. how do we go about doing that? Here I am showing an example where we ahead of time decided to test 96 variants. This number is very much project dependent, cost dependent, depending on your assay. So for some reason, if the throughput is really low, you cannot measure more than 30 or 40, this number could be 30 or 40. If the number goes down, we also reduce the number of substitutions we're going to test. Okay? So in this particular project, we decided to choose 95 because we can have the wild type as a control, making it a nice round figure of 96. And the infologs are designed such that each of these 95 variants are 3 changes from wild type. Those 3 changes coming from the pool of 57 substitutions that we have picked from our phylogenetic analysis of multiple sequence alignment. If you do the math, I chose 57 substitutions because if we put 3 changes in 95 variants, that's 285 different changes, and we have 57 substitutions. What you will end up with is each substitution will be seen in 5 different clones. So our set now is testing all the substitution in a well-represented fashion. There are no random mutations. And also, since they are seen 5 different times with different partners or other substitutions, we can now assess whether a given substitution has purely additive contribution to its activity or does it interact with other substitutions present in the clone. This is critical because what we would really want is a bunch of additive substitutions that we can identify, that we can tag on to the wild type sequence and simply keep getting better activity. So all these sequences that we design are synthesized, and they are codon optimized, and these are all the 95 gene variants have the same backbone DNA as a wild type. So it gives you a very standardized set that we can use to assess the sequence space. And an additional advantage of doing something like this is we can take the sequences that are basically amino acid sequences and transform them mathematically so that we can use a mathematical definition to map the sequence space. Okay, so we started out with the starting point. Then we chose substitutions in a smart fashion, and then we designed systematically varied genes which are based on the original starting point. And now is the next phase, which is really where we're going to put emphasis on, which is testing your 96 clone in a commercial relevant assay. Okay. Since we are only measuring 96, we are going to measure them in what you really care about. And as protein engineers, we all heard of this mantra, which is you get what you screen for. Now we don't have to worry about that because we are screening what we care for, right? So when it may get our K measurements, or if you're interested in plastic degradation, you want to see how well it degrades over time, or if you're looking for a herbicide resistant gene, you can put them in plant, and so on and so forth. Okay? So this is very critical. Our approach now has eliminated the high throughput assay altogether, and we are measuring what we really care about. So once we measured that in low throughput, now it's time to establish the relationship between the sequences and the activities that we have actually measured. Okay? And then it's here where machine learning comes into the picture. As I mentioned before, the infologs can be represented in a matrix form, in a mathematical form, and the activities you measure, I'm going to call them the Y matrix, and they're just in numerical form, it's like a column matrix, and what we call as a model is simply relating the X matrix with the Y matrix in a simple functional form like this. And this is what we call machine learning. There are many algorithms to derive this functional form, and we use simple additive models like partial least square and ridge regression to decipher what this function is. Okay. Now what do we get from doing that? Okay, so here is another example of how I represent the functional form of the sequences associating with the activity. What we get out is weights assigned to each of the substitutions toward the additive contribution or toward the measured activity. So each substitution contributes somewhat toward improving the activity or decreasing the activity of the wild type. And the modeling provides you which one works well and which one does not. For example, N27R, in this project we looked at boosted activity with respect to wild type, whereas P189Q did not. So instead of looking at the best clones, what our approach looks at is the best variables or the best substitutions. But with this information now, you can quickly make a new set of variants, a new set of infologs that has gotten rid of these deleterious or harmful substitutions and started combining good substitutions so that we can march towards our ultimate goal of interest. Now, how do we typically test whether a given model works or not? We do so by following standard cross-validation protocols that are set up by other industries. We simply leave out certain clones and do the model based on the clones that were held by, and then try to predict the clone that we left out. If I can predict those, then my model is good. If I can't, then the model is not good. So here's a simple example where the blue dots show that I'm predicting it, including everybody, and the red dots are showing the cross-validation when I leave out. So obviously my model is slightly poorer with cross-validation than the blue one. Well, in essence, what you get out of it is basically an understanding of what these substitutions bring to the table, what substitutions are good, and looking at engineering based on substitutions, not based on top clones that I get as hits from screens and high throughput screens. Okay, so I'm going to show you one slide from one of the projects that we have published. This is a collaboration with Pfizer, where we engineered an amino transferase over 4 rounds. And the most important aspect I would like to emphasize here is we measured a mere 307 clones total, and we were able to get 200-fold improvement in activity using this approach. And we measured what we really care about. We did not do any surrogates, and even if the assays had noise, I'm going to show you in our tool briefly how we handle it. Here is another example with another corporate partner where we are improving thermostability. So the round 1 infologs that we designed are in blue, and we built a model, and then we built round 2 infologs, and surely we were able to see much better improvements. And our approach being used to engineer several proteins. Here are some published examples. All these links are available on our website. Some of them have open source, so PDFs are available. Some of them you can find them in the library. The most recent one that we're really excited about is this publication in Assay Synthetic Biology, where we basically took the exact same approach that I mentioned today to engineer a herbicide that was resistant, a gene that was resistant to herbicides. And here is a summary slide with several corporate partner projects that we've been working on. And again, some of them are not published, but definitely I want to give you the breadth of different types of projects where we have applied this method. We have used this to engineer thermostability, engineer productivity in a biofuel pathway, in a biosynthetic pathway for intermediate, and also several enzymes in the chemical synthesis pathway for a food and nutrition company. So that brings to the end of my talk, but briefly before we go, I want to show you a little tool that we put together for you to play with to get a better understanding of how noise, substitutions, library complexity affect what you screen. Okay? And you can find that tool on our website. If you go to our services, Protein Engineering, and we have a little link there called Interactive Modeling Tool. If you get there, you find this web page here where it's pretty interactive. So you can read through this to get more information. But the idea is to say, okay, in a perfect world, no deleterious mutation, an average effect of a substitution is neutral. And let's say I want to vary 50 positions, 3 amino acid changes per position. This would be my distribution of activities for the clones. So I'll get a lot of hits, but that's not reality. In reality, if you're making a library, you're probably screening thousands of clones, and your average deleterious mutation in a random library is going to be high, and your average effect of substitution is going to be small. Okay, and it's small negative too, so to speak. And then what you see is still you can pick out hits because what you really screen is what you care for. But that all changes when you start introducing assay noise to the system. Okay, here is when, hey, what if my assay noise is 40%? Now this is the axis you observe. This is the actual activity that you will never observe. Now you see lots of clones here that seems better than real activity, but they are not. And to compound that, you're going to, in high throughput screening, you're going to have a surrogate assay. So you say your assay may be only correlated, let's say, 0.7 with true activity. Now you try to see how your screening is working. You're picking all the stuff that's not truly active. And then also gives you an idea of what the dead clone percentage based on 20% deleterious mutation. But even in this scenario, what we do is we don't look at top clones, we look at top substitutions. And we are not going to make any mistakes here because modeling automatically resolves based on this data what the top substitutions are. Okay? And I let you play around with it, but our typical approach, let's say in a ProteinGPS world, your number of variants are going to be a lot fewer because you're going to measure it in low throughput. And that's primarily because your surrogate is going to be much higher, so you're measuring what you really care for. And if you improve your assay, then your noise will be lower. So all that put together will show you why ProteinGPS is bound to work. And it's very hard using surrogate and high throughput screen to engineer protein. So I'm going to leave it there. This tool is out there on the web. Play around with it and get a better understanding. Thank you.
Great. Thank you so much, Sridhar, and thank you to everyone for joining us today. We hope you've discovered a valuable new set of tools with which to move your research forward quickly and efficiently, and we look forward to working with you on your next project. We'll now answer as many of the questions that remain as possible, and if you have additional questions, you can always reach the DNA 2.0 team by phone or email. As a reminder, if you have a question, you can submit that right now by typing it into the question box on the right side of your screen. So, Sridhar, the first main question that came up several times is, which type of enzymes or antibodies do you think this system is best for?
Actually, that's one of the things I tried to address at the end in the last slide. We have done in many different kinds of systems. We have engineered proteins for thermostability. A lot of our projects has been on enzymatic activity, let's say conversion percentage of A to B or KCAT or KM. And I don't see any limitations. I mean, the idea is if amino acid changes affect activity and you can measure that activity in a true fashion, this method is applicable.
Great, thank you so much. Another question that came up several times was in the website that you were showing, is this actual data or is this, where's the data coming from?
Okay, so I tried to make it clear in the beginning paragraph. These data are simulated. So what I try to do is to say, hey, I have this protein and I'm waiting 57 positions. If I say my deleterious rate is 0, average substitution is, then I assign arbitrarily a weight from a random Gaussian distribution. This is what the amino acid is going to do to your protein, but I don't know that, right? Then what I do is say, take that and then take the protein, make 96 variants, and then simply measure based on the activity that I generated. But really, so the underlying data is simulated in this case, very similar to a lot of projects that we work with. In our ProteinGPS, our deleterious rate of substitutions is 5%. average effect is about minus 0.2, but what we care about is the assay noise. So that is basically any given clone is randomly assigned a noise, and this tells you how related, how observed values are related to actual value when you start transforming those data with noise and surrogates.
Great. A similar question just popped in. It said, how do you calculate assay noise? They assume it's a theoretical value, but how does it correspond to the actual function?
So let's assume that actual function is in this axis, and we don't know what the actual activity is, right? So let's say truly A to B conversion happens in an enzyme, but you are only observing that through some experimental methods that we have. So maybe be it mass spec or whatever that we may use. So those instruments bring in inherently some noise. And secondly, also the reaction conditions. I mean, some are measured in fermenters, large scale, small scale. They have inherent noise that they bring in. So what you actually observe is not the true activity. So noise here I refer to as, if I take the true one and I transform them, each of those randomly at 20% Gaussian noise, then that's what I observe. And same goes for surrogate correlation.
Great, thank you. Another question that was several people asked is, how do you deal with the interactions between amino acid substitutions at multiple sites in the analysis?
So in our analysis, to pick the substitutions, we are not looking for amino acids that are covariant, for example. Okay, so we just pick amino acid substitution based on the assumption that most amino acids are not interacting with something else. So what I want to show you is what our model does is also assuming that amino acid substitutions are additive and not interacting. So if you put non-additive substitutions into this model, so let's say a given substitution is it depends entirely on what else is present, then remember we are seeing that substitution 5 times. So a couple of times it may be seen that somebody likes, 3 other times it may be seen that somebody doesn't like. But the model is looking at an average, so it will bring the contribution brought by that clone, by the dependency, to be somewhat neutral or negative or maybe slightly positive, but not the top. So this model automatically resolves them. And to an extent, I will be missing them, but I don't really want to get involved with substitutions that are going to affect my function by new substitutions that I'm going to bring later on. I really want to focus on additive substitution. By assuming that things are additive and building a model assuming things are additive, the non-additive substitutions get left behind as we go from one round to the other.
Great, thank you. Another interesting question, wanted to know, how will the final outcome be different with a different number of mutations? For example, 3 mutations per variant, or 4 mutations per variant, 1 mutation per variant?
Yes, so the model, as you can definitely look at it, I accounted for that in the tool. Try that and see what it says. The model does have certain requirements. We cannot have thousands of variables and only tens of clones because I don't have enough variables. So it's just a classical fitting problem, you know. You want the clone and the number of substitutions in each clone to be limited, and also variables to be limited. So certain combinations will not yield models at all because we are not sampling it enough. So they are all related, but you can look at the tool and have certain conditions put in there. When you choose too many substitutions or too many varying positions and too few clones, it will say I can't model. And also the number of substitutions per variant is sort of critical in the beginning, because if you increase the number of deleterious mutations and if you put in too many substitutions in a given clone, it's bound to have one of those deleterious substitutions in it. So you want to limit that too.
Excellent. Thank you so much. An interesting question that a few people ask is how successful is ProteinGPS? You know, how frequently does it actually identify better functioning proteins, increase whatever people are looking for. They're just curious, what's your success rate?
So we have done over 25 projects, and pretty much every time we found something better. And sometimes the goals are lofty. We may not have reached the ultimate goal, but in every case we have improved from the starting point. Okay? In most cases we reach the end point because oftentimes we are not just engineering something better and better and better. There is a commercial endpoint goal, which is often set by downstream processes. You know, if I get it to this activity, then I know I can make something commercially relevant. So that's really what we aim for most of the time, not something really, really good. So sometimes we haven't reached that goal, but we have improved and approached that goal.
Excellent. Another question I think is interesting is this an efficient approach for assaying properties of selected library members?
It is, but the modeling works best if the data is systematically varied. When you do random libraries, some substitutions may be well underrepresented and some may be overrepresented, and we may not gain any knowledge on the variants or substitutions that are underrepresented. So, if you want to measure only 100, you want the best out of it, and it's best to build such an influx set rather than sampling from a random library.
Excellent. I think we have time for maybe one last question. And it's sort of a general question that several people asked, which was, what would be the process for starting a ProteinGPS project? What do you recommend?
Well, I mean, definitely you can get in touch with us. Typically, processes, a lot of our customers have endpoint that they want to achieve. They have a starting enzyme that might work. They may be looking for a better starting point. Just a matter of engaging with us, if that's what the question is. How do you work with us? Yeah. So, and then we go off setting up a little conference call or something like that. You know, try to understand more about what you're looking for and help you out.
And as an addendum, someone else just wanted to ask, and I'll throw this in as the very last question, about how long does the process usually last to do a ProteinGPS project? Is it a few weeks, months, years?
Yeah, sure. That's an easy one to answer. So the computational part of designing is not much. It's about a week to start with a gene and then design them, and about a couple of weeks, maybe 3 weeks for us to actually synthesize them, you know, going from virtual to physical gene. So you're looking at about, for every round, the modeling and synthesis is about 3 to 4 weeks, and whatever time it takes to assay 96 is the remaining time. So you can add that and multiple. Typically we've done 3 to 4 rounds per project, not more. We have done some projects with 11 rounds, but some with 2 rounds. You know, we got something better already. So you can just say, if your assay time to assay these is 1 month, for us to synthesize and model is 1 month, you're looking at 2 months per round, and you can do 4 rounds, you're looking at a year or less for a typical project. But on the other hand, if you're growing corn, it might take long.
Excellent. Thank you so much, Sridhar, and thank you to all the researchers who joined us today. We hope you've discovered a valuable new set of tools with which to move your research forward quickly and efficiently, and we look forward to working with you on your next project. If you have any additional questions or the questions that have not been answered yet during the webinar, you can contact us by phone or email as noted on the last slide. A reminder, a copy of the webinar and the slides will be available on our website in about 24 hours, and any questions we weren't able to get to this morning we will answer by email. For more information on DNA 2.0 products and services, you can visit our website, and thank you so much.